Hello everybody, welcome to the unit on sugars. This is lecture 17A. Let's keep it sweet and simple. So I'm going to begin by giving you a statistic that I like to quote a lot. It just gets at the heart of why carbohydrates are so incredible, why they're so important. They are 85% of everything that's alive around you if you were to remove the water. 85% of the dry biomass on Earth is made up of some sort of a carbohydrate. Admittedly, most of that is cellulose fiber, but certainly by no means all of it. Carbohydrates themselves are coincident with life on Earth. They are prebiotic. We had to have carbohydrates in place for life to begin on Earth. They were the first category of biomolecules to begin. Basically, Earth was a giant pile of rocks and water with the sun beating down it. It was sterile. There were no biofilms, there were no bacteria, no microorganisms. There was nothing, just rocks and water and sunlight. The first carbohydrates happened through the action of what are called the banded iron formations. There were these iron deposits in these rocks that catalyzed this. They were part of these redox cycles that enabled carbon dioxide to get fixed into very simple carbon-containing molecules that then were able to combine into larger carbohydrates. So acetaldehyde and the larger carbohydrates came out of this. This is work that was first established back in 2000. So we've known for about 20 years now that carbohydrates really were the first biomolecule on Earth before biology, and what the mechanism of their formation was. Carbohydrate as a category are molecules that have a carbon that is hydrated. So a carbon, hydrogen, and oxygen in a 1:2:1 ratio. That is carbon, CH2O. And then subscript N, meaning that the carbon chain can be any variable length. The most common value for N is 6, and we'll see that in a little bit. But right here, this is the simplest of the sugars, is glyceraldehyde. So a monosaccharide, or a simple sugar, is just the simple single molecular unit of a carbohydrate. On the left, we have glycerol. We ran into glycerol when we were talking about triglycerides, and how the fatty acids would form esters with the 3 hydroxyl groups in glycerol. If you were to take glycerol and oxidize one of the hydroxyl groups to an aldehyde, to a carbonyl group, that's called an aldehyde there. This gives us glyceraldehyde. That is the simplest sugar. So a sugar is going to be a molecule that has a chain of carbons that each have a hydroxyl group attached to them, and one carbon that's going to have a carbonyl group, either an aldehyde or a ketone. So that's the simplest sugar, glyceraldehyde, right there. And its empirical formula is CH2O subscript 3. This is the tree of aldoses, that is carbohydrates. When you see that suffix OSE, it means a molecule that is some sort of a sugar. Aldoses are sugars that are based off of aldehydes, like we saw in glyceraldehyde. And this is the tree of complexity that grows as you add carbons onto that glyceraldehyde chain. A triose is a 3-carbon sugar, tetrose 4, pentose, and hexose is 6. These monosaccharides are simply the monomeric carbohydrates that get categorized based on the number of carbons in this aldose tree. And each of them in a certain level of this tree, meaning that they have the same number of carbons, are all isomers of one another. So they're multiple compounds that have the same formula but a different arrangement of atoms in the molecule. When you look at, for example, in all of the hexoses there, we have allose, altrose, glucose, mannose, gulose, idose, galactose, talose, etc. Each of those varies in the relative stereochemistry or configuration of the hydroxyl groups about the various carbons along the backbone. The four groups around any particular carbon are each different, and there are two different ways that can be arranged for each of those what are called stereogenic centers, or each of those carbon centers. And that's what gives us the different identities of sugar molecules. These are the common ones, the very common ones. So all of these are possible, and more than this certainly do occur in nature, but these are the ones that you're going to run into commonly. Glyceraldehyde is part of glycolysis. You see it in all sorts of contexts. Ribose is the sugar that is in RNA, and the deoxyribose is in DNA. So that's very common. Glucose is blood sugar. Mannose and galactose are both common sugars found in more complex carbohydrates in larger structures that do all kinds of things in the body. So all of those are very common sugars that you'll run into. So let's look at the categories of sugars and their polymers. Monosaccharide, these are the simple sugars, and there's 20 of the known 70 or so monosaccharides occur in nature. So that means there's about 70 different molecules of monosaccharides, and approximately 20 of them can be found in natural contexts. And here's an example of, what is that, fructose. That would be high fructose corn syrup in that picture. A disaccharide is when you link two of these together. So that's a molecule that's formed by what's called the glycosidic linkage of two monosaccharides. We'll see that in the next lecture, what that actually means. But some examples of that are sucrose, or white table sugar, and lactose, which is the sugar that's found in milk. And then polysaccharides are larger macromolecules made of long chains of these monosaccharide monomers that get linked up in all kinds of either straight or branched chains. So there's all kinds of examples: vegetable starch, animal starch, which is also called glycogen, soluble and insoluble fibers. I'll talk about all of those in later lectures here. So just a quick note on the structural formalisms of drawing carbohydrates. Carbohydrates are able to be in either a closed chain or open chain form, and you'll see them drawn in either way. The open chain form is called the Fischer projection. And it turns out this guy, Emil Fischer, was actually my academic great-grandfather. The PhD advisor of my PhD advisor of my PhD advisor was that guy. And then the closed ring form that you see here, that drawing was first come up with by this guy, Norman Hayworth, and we call that the Hayworth projection. So essentially, when you make a ring, the hydroxyls that are on the upside of the ring are all the groups or substituents that are pointing upwards relative to the ring, and then all the ones downward are, of course, on the other side of the ring. Is there anything else here? Yeah. The guy himself, Hayworth, was a pretty interesting guy. Got the Nobel Prize for studying carbohydrates. He was also the person who elucidated the vitamin C structure. Pretty interesting chemist. Also got the structure of maltose, cellobiose, which is the dimer, the disaccharide that's part of wood pulp, and then lactose, milk sugar, etc. Did a lot, carbohydrate chemist. All right, let's keep moving though. So when you look at one of these carbohydrates in its circular or ring form, you'll notice that one of the carbons has 2 bonds to oxygen. It's bound to 2 oxygens. This is what we call carbon 1. It's also called the anomeric carbon. So in this model here, which is a picture of glucose, we just start from carbon 1 and then we go down the carbon chain working along the background, the backbone all the way to carbon 6. That region that has the carbon that's attached to the 2 oxygens, that's a special part of the molecule. We call that the hemiacetal functional group. It has some important chemical significance, which we're actually going to explore only insofar as we talk about it in its relationship to ring opening. This is the site of ring opening when the carbohydrate becomes an open chain. Here are some other ways of just looking at a picture of the same molecule. This is all beta-D glucose. So we have the Fischer projection using a closed ring form of the Fischer projection, a Mills projection, which is something that's looking down right on top of it, the Hayworth projections looking at it from sort of an angle from the side, and then this Hayworth projection from the chair conformation, also looking at the side, is probably the most common. It's my favorite. This fourth method here is my favorite way of drawing the molecule. So there are 2 directions when talking about the structure of a carbohydrate. There's axial, which is up and down straight through the ring, and then equatorial, which is in the plane of the ring. And you can use that language to describe the orientation of the hydroxyl groups. In glucose, in alpha glucose, which is described in that little box there, you have 3 equatorial hydroxyls, and then the anomeric hydroxyl at carbon 1 is actually axial in that orientation. If it was beta, it would be a equatorial hydroxyl at that carbon. All right, so I've been using this language here. Let's define it. Anomers are simply for a given molecule of a particular sugar, that closed ring form can open and then close again. But as it closes, it can close one of 2 ways with the configuration of that hydroxyl, either axial or equatorial. So because of that, when you make a solution up of any of these what are called reducing sugars, those sugars are going to be present in a mixture of these anomers. So there's the alpha anomer and the beta anomer. Alpha anomer has the hydroxyl pointing axially, and beta has it pointing equatorially or above the ring in the Hayworth projection. Anomers are otherwise totally identical sugars that are going to differ in their configuration of the carbon attached to both oxygens, the anomeric carbon, in the hemiacetal or hemiketal if it's in fructose or keto sugars. The difference is only important when you think about glucose polymers. If you were to take, if you were somehow able to get pure alpha-D glucose, I don't know that it would taste any different than beta-D glucose. Anyways, you can't get it pure because alpha-D glucose, when it's in solution, comes to equilibrium with actually 64% of it. It's going to be 2/3 of it is beta, and then only about 1/3 of it is the alpha. All right, so now that I've completely confounded you with some high-level carbohydrate chemistry there, not that high level, you can handle it, I want to compare a couple of the most common sugars. I told you there were 20 naturally occurring, 70 possible. There's a lot of space in the structural biology of carbohydrates. We're just going to explore the 2 most common monosaccharides. Glucose, which is that hexoaldose, and then fructose, which is actually not an aldose sugar. It's a ketose sugar because instead of having its the carbonyl group at C1, it's at C2. And that leads fructose to be a ketone rather than an aldehyde, and it forms this 5-membered ring rather than a 6-membered ring that you see in glucose. So fructose is fruit sugar and glucose is blood sugar. Yeah, both monosaccharides. We're going to compare the two of them a little bit. So, whoa, heavy slide. No, not that bad. It's okay. So here we are. We have, we're going to explore how each of these sugars interconvert between the open chain and the ring forms. So we have on the top row here glucose, and we have it in what looks like the beta form, beta D-glucose in a 6-membered ring, and it can be in equilibrium with the open chain, where the hemiacetal goes back and forth between that, the hemiacetal, and the aldehyde and hydroxyl group of the open chain. And this is just a dehydration reaction. Fructose, on the other hand, so you can see here in the open chain form that carbon 1 that has the double bond to an oxygen, it is that carbon has one bond to another carbon and another bond to a hydrogen. That's what makes it an aldehyde. A C double bond O, where that carbon is also bound to a hydrogen, is what we call an aldehyde. And it has slightly different chemistry than when that carbon bearing the double bond to an oxygen is, the other two bonds are both carbon. That's what makes a difference between aldose, a sugar being an aldose, versus a ketose. So fructose is able to open to form a ketone in very similar chemistry. It's identical chemistry to what happens in the opening of glucose. The only difference is in the position of that aldehyde group in the carbon backbone of the glucose versus the fructose. All right, so there was some pretty intense chemistry for most of you, I imagine. So I'm going to put up a picture of a cute puppy dog who's kind of confused here. He's going to try to follow this. The next concept I am lumping into this lecture is the idea, perhaps something maybe a little more relevant to you, sweetness index versus glycemic index. So sweetness, and these two ideas are often confused by people, and I'm going to explain them and the relationship between the two of them. This should be something very practical and useful to your life. The sweetness index is, well, first of all, sweetness itself is just one of the gustatory sensory modalities. It's one of the 5 types of taste that you have in your body. The sweetness index is just a relative measurement of how sweet we perceive a fixed amount of something to be relative to the same amount of some other thing. Okay, so I have 2 tablespoons of 2 different types of sugar. Stick one in my mouth, how sweet is that? Stick another in my mouth, how sweet is that? Sweetness index is just a super crude relative measurement of how sweet one is with respect to the other. The glycemic index, on the other hand, is a measurement of how rapidly a fixed quantity of food spikes your blood sugar. So we had those 2 sugars that I was talking about before. We take a tablespoon of one and a tablespoon of the other, and I eat each of them at different times of the day. They'll have different glycemic indexes or indices. The one that has the high glycemic index is going to be the one that spikes the glucose that ends up in my blood very rapidly, and the one with a low glycemic index is going to be the one that's converted into glucose in my blood very slowly. So they both may deliver the same calories to my body, but one is just spiking the glucose much more rapidly, and the other one is spreading it out over a longer period of time, maybe because it's harder to convert into glucose or something like that. So we're going to explore those ideas here forthwith. Say goodbye to the cute puppy. All right, let's compare alpha glucose with fructose. So glucose syrup is something that you commonly see in the store. It's often used in baking. It makes your baked goods a little bit more moist without adding a huge amount of sweetness to them. You can also use it, some people use it in drinks, cocktails. So which one is sweeter, glucose syrup or fructose syrup? Here's some high fructose corn syrup that was isolated from corn. Basically pure glucose on one hand and pure fructose on the other. Get them right off the shelf at the local grocery store. Which one is sweeter? You stick a tablespoon in each and stick them in your mouth. Which one do you think is going to taste sweeter? Well, it turns out that fructose is actually about twice as sweet as glucose, just on this relative scale. By definition, we call sucrose 100. We just call sucrose 100 for just as an arbitrary definition. Fructose is more sweet than sucrose. In this relative scale here, it's about 140 on that index, where glucose is only about 70. So fructose is twice the sweetness as glucose. All right, so that's the basics of sweetness index. Let's move on to glycemic index. This is just a number from 0 to 100 that we assign to a food. It actually goes above 100. Assigned to a food with pure glucose, again, arbitrarily given the value of 100. This represents the relative rise in the blood glucose level 2 hours after consuming that food. And so notice in the previous example we used sucrose as the definition. Here we're using glucose, two different sugars. The ability of a particular sugar to cause a spike in your blood sugar is dependent on a lot of different factors. So the quantity and type of the carbohydrate that food contains, the amount of entrapment that the carbohydrate molecules within the food. So for example, is that sugar that's in the food that you're eating, is it traveling with a lot of fiber, maybe soluble or insoluble fiber? And if it is, soluble or insoluble fiber tend to make it a little bit more difficult to extract the sugar from the food. This is why it's good to have, if you're going to consume fructose, have it in the fruit that it originally came in, because it is much less detrimental to your blood sugar. And then the relative amounts of fat and protein in the food are also going to affect the glycemic index of that particular food. There are various organic acids or salts in the food. And then if the food is cooked, and if so, how was it cooked, that's going to make the carbohydrates, the starches in that food, more or less accessible, and is going to affect how rapidly they're converted into glucose in your blood. So all those factors can affect the glycemic index of a particular food. Here's just a chart of the different sugars and foods and what their glycemic index are of grains, vegetables, fruits, dairy, and proteins. You can look across that. And you'll notice that the really high numbers exist in the grains and starches. There's a couple high numbers in the dairy, but that's really because there's added sugar to those, like ice cream. We said that glucose syrup is by definition 100, but fructose, let's look at how widely that number can vary. If you were to eat fructose, just say in a piece of fruit, like you ate a strawberry or something, the glycemic index of fructose in that in the context of fruit may only be 19, like very low glycemic index, whereas it can be upwards of 70 or 80 if you were to take a tablespoon of high fructose corn syrup. So still lower than glucose, and that's because the body has to convert the fructose into glucose to be able to have it have a measurable effect on the glucose in your bloodstream, and that takes some time, so that kind of slows the whole process down. So you may be asking, why the hell does this matter? Well, let me tell you. The glycemic index is important because foods that have a high glycemic index can stimulate a strong insulin response. And if this happens over and over, this is how you get insulin resistance, type 2 diabetes, all that nasty stuff. So you don't really want to consistently eat foods with high glycemic indices, because that leads to like chronic spiking, really intense insulin reactions that down the road can lead to type 2 diabetes. Sweetness index is important because the sweetness of a food doesn't necessarily need to correlate with its glycemic index. So you can have a food that doesn't actually taste very sweet, but it has a really high glycemic index. So the classic example of this are starchy foods. So here are a couple foods that have very low sweetness index. They're not sweet. If you eat a potato or white bread, you don't think to yourself, geez, that's really sweet. But they both have extremely high glycemic indices. Potato is 85. White bread is basically solid glucose of a glycemic index of 100. And let's compare that to what Homer's doing here, taking a grapefruit and jamming it into the sugar bowl. Grapefruit is only 25 as a glycemic index, actually a pretty good food to eat, and that's because of the entrapment of the fructose that's in the grapefruit with the fiber that that comes along with the fruit sugar. And the sucrose itself has a pretty low glycemic index as well compared to potatoes or white bread. And that's because sucrose is a disaccharide made up of fructose and glucose bound together. The body has to go through the process of breaking that apart into the individuals, and then half of that sugar, the fructose, needs to be converted over to glucose. So even though a grapefruit jammed in the sugar bowl obviously tastes much sweeter, and you think that it might lead to a greater spiking of the blood sugar, actually eating a breakfast of hash browns and white bread toast is going to be a faster spike in your blood sugar, a higher glycemic index. So that's why all those concepts matter. It's important to understand the relationship between sweetness and glycemic index. Okay, thank you for that. Sticking through that lecture. Stay tuned for part B up next.